Hello, hello, hello. Okay, totally impromptu session, this one. I didn't get a chance to jump online and do any questions with YouTube last night, which was my live class night, normally on Thursdays, and I was just feeling a bit guilty about that. I thought I'd jump in. I've been getting quite a lot of questions about prokaryotes and eukaryotes and stuff like that, so I thought I would just sort of share a question with you. If you've got any A-level biology questions, chuck them in there. And let me know. I can answer them. I'm going to just take you through this question. I've seen it answered pretty badly in the past, but it's a pretty easy one to figure out what the question is going on about. But yeah, I'm going to just walk you through it and show you how you can sort of tackle it to get full marks on the question, basically. Another thing, just like I would say, general point of fact, we do YouTube lives like this kind of off the cuff quite a lot of the time. So hit the bell and make sure you don't miss out on any of the stuff that we're doing for free on YouTube. Right now, we're also running like a free trial, so you can check out all our courses for free. If you've got any questions about how you can use Tailored Tutors for a month for free, just ask us. I can share the link to you as well. It's the free trial page on our website. But okay, so I'm going to jump in and just delve into this question, break it down a little bit for you. If you've got any suggestions for what you want in terms of, in terms of guidance in through the answer, a lot of A-level biology is about figuring out what the question's about, how to break it down so that you know what to write for the answer. The knowledge is obviously something you're going to have to learn, but the skill for breaking down the question is super duper duper important. So let me just run through this one just nice and quickly. Okay, so let me share my iPad with you here. Yeah, okay, so the question that I've got here is from an old spec paper. It's still totally relevant. With all my classes, I'll select sort of either new spec questions or old spec questions, but they're all going to be relevant for AQA, OCR, and EdXL students within my course. So, okay, so let's just analyze the question. If you followed my exam guide at all, I was analyze the question in blue. So the structure of a cholera bacterium, and we're talking about the structure, is different from the structure of an epithelial cell of the small intestine. Our command word is describe how the structure of the cholera bacterium is different. So first thing I try and train my students to do is rephrase the question. Let's strip away all the nonsense here. We've got 5 marks to get. So this is describe. The command word is not allowed to change, but describe is an AO1, so factual recall. The structural, because that's important because it's in the question, differences between. Now, any suggestions? I'm not going to hang around too long tonight, but you got any suggestions as to how can we simplify this question to make it a little bit easier? Between what? Well, we could say cholera bacterium, we could say epithelial cell. What's it really want? What's the mark scheme going to tell us here? Well, the structural differences are going to be between basically a prokaryotic cell and a eukaryotic cell. So that's our question. That's really what we're supposed to be answering here. The fact that the fact that the question doesn't tell us that directly, that's one of the skills you got to learn, how to break down skills. And it's one of the main things I can teach you in my live classes is how to open out these questions, so just simplify them so you kind of know what to write. So I'm just going to say, what would I be thinking here? Okay, well, I've got 5 marks. So one of the things we can do is like, what are the main we don't want to know what they have both. We're not going to talk about cytoplasm. They both have cytoplasm. All those things that they both have. If it was a compare and contrast question, suddenly now I can talk about the things they've got in common. They both have cytoplasm. They both have a cell membrane. But this is only looking for the differences. And so we can't do that in this question. We've got to give the differences, and you've got to make it nice and clear which ones have which. You can use bullet points. So I'm going to switch to black for my bullet point answers. What am I going to say basically for any suggestions? If you've got them, throw them in. I'm going to say cholera is a bacteria, or bacteria are prokaryotes. They have a circular loop of DNA with no nucleus, and no histones, which are the proteins around which the DNA folds in a eukaryote. So, I mean, that's basically probably all I need to say. We could maybe really stamp it home and say a eukaryote or the epithelial cell has a nucleus. It has linear DNA. You're welcome to do that if you want to. I'll show you the mark scheme towards the end. Maybe I'll do it for just to really make it clear from my teaching perspective. The epithelial cell so that linear DNA with histones in a nucleus. Remember, on the exam, don't write out here. If you've got that line that says don't write outside this line, don't write outside that line, because it will not get scanned and it will not get seen, and therefore it will not get marked. Okay, so I've probably written more than I need to there, but that's like full detail just to give you all the sort of the backstory. We can say that the epithelial cell has mitochondria, and what would I'm going to write Golgi if I can spell it correctly. Capital G for Golgi. You can call it a Golgi body. You can call it a Golgi apparatus. Prokaryote does not. But the bacteria or the cholera does not. So that's things that the eukaryote has that the prokaryote doesn't. And now can you go the other way around? So we can say the bacteria has, can you name any? Probably you can think of a few features. So we can maybe talk about a slime capsule. We could talk about pili. We could talk about plasmids. Some of the features which are unique to prokaryotes and not eukaryotes. I'm going to go with a cell wall. And if you want, you can tell them what the cell wall is made of here. They'll accept two things, murin or peptidoglycan. And plasmids. And the epithelial cell does not. Okay, so I've got how many features here? I've got plasmid, cell wall is 2 features. I've got mitochondria and Golgi body is 2 features. I've got linear DNA versus circular DNA. I've got histones, which might be a mark in there as well, but 1, 2, 3, 4, 5. I'm confident I've got my 5 marks. What I'm going to show you next on here, this is in the Taylor Tutors free trial. So if you go to the Taylor Tutors website and you're looking at I happen to be in the AQA version of the course right here, but you can essentially take a look through and you'll see all these, can I draw? No, how do I actually move around on this thing? I tried to be fancy and embed a browser into my iPad as a bit of an interface, but I actually don't know how I move around within it. Okay, but you've got all the sections down here. These padlocks and the unlocked padlocks just show us that you've got free content in each of those sections. So you can drop these down. He says, it might just be easier if I just share my screen on my computer instead. Can I do that? Yeah, okay. So you can then see the ones that are not grayed out are the ones that are free. If you want, you can also look at the course overview page and you'll see all of these sections. Obviously, we're currently looking at prokaryotes and eukaryotes, so you can tap on that and it will take you down to the section. It'll then also show you all the lessons that are in here, and we're looking at prokaryotes versus eukaryotes, which would be this lesson just here. And it'll then take us back there. So if you watch the video, in fact, what I can do is just skip to the end of the video. I'm not going to play the whole thing. Obviously, it's all in the course. But you can see how that table is laid out. Maybe if I can make that full screen, it will just make it a little bit clearer for you that this is how my lessons are structured. I color code everything. And you'll be able to see all the differences between the prokaryotes and eukaryotics in how long is this video? This video is 7 minutes. Well, I'm at 7:17 and there's 15 seconds left to go. So under 8 minutes for the video. And And then, in fact, what I can do is also, I can just show you the mark scheme for this question as well. So for the mark scheme here, what have we got? We've got one for stating it's a prokaryote. So that gives us a mark, which does not have a nucleus, nuclear envelope, DNA is free in the cytoplasm or a loop of DNA. You're allowed 2 marks, any 2 from membrane-bound organelles, mitochondria, Golgi, endoplasmic reticulum, etc. So any of the other membrane-bound organelles will be present in the eukaryote, but not in the prokaryote. You could say small ribosomes only. I didn't mention that because they both have ribosomes. They are slightly different types, 70S, 80S, small or large. You can use either of those. And marking points 6 and 7 available, capsule, flagellum, plasmid, cell wall, etc. So a maximum of 2 marks for these guys down the bottom, and a maximum of 2 marks for any of these. You should always mention 2 of the organelles if you're asking to compare the differences between prokaryote and eukaryote. And yeah, I mean, that's just a nice summary of both of those things. It's kind of a question that is frequently quite poorly answered, mostly because of the mystery in the question rather than the actual like difficulty of the subject matter as such. So yeah, that was the thing that I was going to be doing with you on Thursday, but I ran out of time because I was actually planning my live class. I wanted to make sure I had enough question examples that were like really on point for my students. So if you want to join me in those live sessions, in fact, what I can do right now, I can just show you, where is it on the website? I will show you. I'll find it and I'll show you. Okay, so I'll just share my computer desktop right now and show you the Tailored Tutors website. And if you do want to ask me questions on any of this, I don't answer questions about biology content on YouTube. It's just there's thousands of students who are asking me questions there. Simply, there's not enough hours in the day. I answer all my A-level biology students inside the academy. And right now you can ask me questions and it won't cost you anything. You can try out the courses. I'm going to show you how to do that from our website. So if I just share my screen with you, you've got all our subjects in the menu here. It's probably easiest if I'm just going to type it in to free-trial. And you can go here. In fact, you can also click on that big fat button on the homepage. Now I look at it. And this will take you to a page. It will tell you all the information about the coupon to use. It will tell you the information about how you can, there's a little video here. That's me. You can just, here's the coupon code. So you can use the coupon code TryTT-free. You will need to start a subscription, use your card details, all that kind of stuff. But yeah, if you don't want to continue it, then you don't have to. You can cancel it anytime in the first 29 days. So well worth jumping on. You can then sort of jump inside the courses, see what they're all like. Yeah, that is the easiest way to basically come and ask me questions about any of the A-level biology content that your heart desires. Okay, so I think pretty much that's more or less it from me. Yeah, and normally I'll be back here Thursdays. As I said, we do pretty random YouTube live sessions. I've got a few more YouTube live sessions coming up for you guys in year 12 and year 13. So hit the bell and subscribe so that you don't miss any of them, basically. And yeah, if you do have any questions, send them through to us on the channel. Get in touch with us via the website. And yeah, we'd love to see you inside the courses. Take it easy. Have a nice weekend.